Yep.
All right. Well, good morning, everyone, and welcome to today's Grand Rounds. We're very excited to welcome Dr. Michael Levy, who's joining us for the first time post-pandemic from the Ether Dome, hopefully as a sign of things to come. Dr. Levy is an associate professor of neurology here at MGH, and he recently joined our faculty, leading the research unit in the new division of neuroimmunology. His mission is to build a combined clinical and research neuroimmunology program to develop therapies for patients with autoimmune diseases of the central nervous system. Dr. Levy moved to us from Baltimore, Maryland, where he was on faculty at Johns Hopkins since 2009 and director of the NMO clinic there. Clinically, he specializes in taking care of children and adults with rare neuroimmunological diseases, including neuromyelitis optica, transverse myelitis, MOG antibody disease, and acute disseminated encephalomyelitis. And he is the principal investigator on several clinical studies and drug trials for these conditions. In addition to editing neuroimmunology reports, Dr. Levy is a co-chief editor at Multiple Sclerosis and Related Disorders, as well as a member of the editorial boards at the Journal of Neurological Sciences and Scientific Reports. In his brief time at MGH, he's already demonstrated his commitment to trainee mentorship, and he has welcomed multiple residents interested in contributing to his ongoing research efforts, many of whom are joining us today. So welcome, Dr. Levy, and we look forward to hearing your 3 short stories in neuroimmunology.
Thank you, Priya. It's really an honor to be here. Let's see. Okay. So, I just came, as Priya said, in 2019, and I haven't really had a chance to meet people with the move and then with coronavirus. And Priya gave a wonderful professional introduction, but I thought I would include a little bit of personal introduction about me throughout this talk. And the way I've divided up the talk is into 3 short stories. Most people are signing in by Zoom, and I feel like my attention span by Zoom is about 10 to 15 minutes maximum. And then I need a break, and then I can probably do 2 or 3 of those. So if you're like me, I think you would appreciate this format. I'm going to talk about 3 short stories and then include a little bit about myself. And then, as Priya said, I am live casting from the Ether Dome. When I was invited, I was really honored to accept the invitation to give Grand Rounds here, but I asked for the latest possible date so that I could potentially stand in this room and deliver the talk. And I think things are getting better, but thanks to Carol, made this possible that I could give this talk here with a few people in the audience, mostly from my lab and from the inpatient service. Thank you, Marcelo and Dr. Samuels joining us. So these are my parents. My mom was raised in Paris after the World War II. My dad's Israeli, and I was born in Israel, and we lived in Iran and Israel kind of back and forth until after the revolution. Then we moved to the US, and then I became a citizen at age 10. And I grew up in Houston and went to Baylor in Houston, met my wife. She's from Louisiana. She's pictured here. She's a hematopathologist. We met in med school. And she's much more accomplished than I am. She's the chair of pathology at Dana-Farber, and she has 3 R01s at the same time. Very accomplished, as I said. And then here are my 3 kids, Allie, Megan, and Josh. And they, when they moved here, they really fell in love with Martha's Vineyards. So we've been there many times now on the beach there. Okay, so the title of my first short story is called Never Go Against a Sicilian. And if you're a Princess Bride fan, you know the rest is when death is on the line, right? But the Sicilian in this case is none other than our own Dr. Marty Samuels, who kindly joined us today in the audience. And fortunately, death was not on the line, but a lobster roll was. And the bet was concerning a case of bilateral optic neuritis. This is an MRI of someone with bilateral optic neuritis. We could see the enhancement in the optic nerves from the retina all the way down to into the skull. And the patient that Dr. Samuels and I were talking about tested positive for the MOG antibody, the myelin oligodendrocyte glycoprotein antibody. And so the question came up, well, does this really fit the diagnosis? MOG antibody testing has only been available in the US since 2017. We don't even have clinical criteria for this disorder. What are we really testing, and what is the full spectrum of MOG antibody disease, and where did it come from? It came from the common laboratory mouse. what people used to do back in the 50s, 60s, and 70s was they would homogenize spinal cord tissue and then inject it under the skin of a naive mouse. And by the process of molecular mimicry, the host, the naive mouse's immune system would confuse its own spinal cord proteins with the ones that were injected that were just slightly foreign. And it would cause an ascending paralysis that looked for all the world like a demyelinating disease like MS. And so for decades, this mouse was used to develop MS drugs, and several of them were developed with this mouse model. And among all of the spinal cord proteins, myelin oligodendrocyte glycoprotein is one of the most immunogenic proteins. In the 80s and 90s, they figured out exactly which epitope, which ones stimulated B cell production with antibodies, which ones were more T cell stimulating. And as I mentioned, this idea of MOG as being the primary antigen for demyelinating disease really caught on. And my colleagues in Austria were so convinced that MOG was the target in multiple sclerosis that they published this paper in 2003. This was a big deal in New England Journal of Medicine. They claimed that if a person comes in with a first demyelinating event and they demonstrate antibodies to myelin, especially MOG antibodies, that was a predictor of multiple sclerosis. So this would be the first blood test for MS. It's a big deal. Every lab tried to reproduce it. Every lab that's interested in MS tried to reproduce it, and nobody could. Okay, and so by 2007, there was a new New England Journal paper. The title of the 2003 one is Anti-Myelin Antibodies as a Predictor of MS. The title of the second paper was Lack of Association Between Myelin Antibodies and Progression to MS. So what happened here? What was this failure with this MOG antibody? Well, as I mentioned, almost every lab that's interested in this condition started looking for MOG antibodies. And they found a few. What I'm showing here is every study that demonstrated that looked for these MOG antibodies, they found a few. Here's the sensitivity. And for the most part, less than 5% or so would test positive for the antibody. And this is by a Western blot assay. Don't mind my Australian colleagues here who are particularly excited about this test. Most of them, you can see the average here, less than 5%. And the real problem was that in many populations, there was a very high false positive rate. So up to 10% of Italians, for example, might test positive for this test. And so 5% of MS patients test positive, but 10% that doesn't make any sense, right? It makes the test clinically not useful at all. Fortunately, the British saved us. And what they figured out is that they could improve this test and make it more specific for demyelinating disease, take out all those false positives if they made two critical adjustments. The first is that you have to use a cell-based assay. So what is a cell-based assay? You take a HEK cell or some other immortalized cell, transfect it with MOG so it's expressed on the membrane, and then you add the patient's serum to that culture. It'll bind, and then you add a secondary antibody to detect the human antibody. So cell-based assay number one was critical. Previous Western blot assay, we use denatured MOG, that doesn't work. ELISA testing, we use pieces of MOG, that doesn't work, too many false positives. So you have to use a cell-based assay. Then the British said, not only should you use a cell-based assay, but you have to look specifically for the IgG1 subtype antibody. Okay, so for example, just to remind you what different kind of antibodies you have in your blood, you have IgMs, those are acute phase. You have IgA in the mucous membranes, you have IgG floating around your body, and there are 4 subtypes of IgG. IgG1 is among the most pathogenic. Okay, so what you want to find is the IgG1. So look at patient 1 here. Patient 1, patient's serum was washed over. They used a secondary antibody that's used in Western blots, for example, that binds to any part of the heavy or light chain of IgG, and this person tests positive. Okay, but then when you add a secondary antibody to IgM, they also test positive. So they have IgM antibodies to MOG, but this patient also has IgG1 antibodies to MOG. So this patient is a true positive. The second patient has a little bit of positivity here to the non-specific secondary, but only has IgM. Okay, and this patient is a false positive, okay? And then the third patient here has a positive test with the non-specific secondary, no IgMs, does have an IgG1, that is another true positive. And then this bottom case are all the controls in Italy, for example, and actually this is also me. So I harbor IgM antibodies to MOG. I found out when I was helping to develop this test that this is me, this is what I would look like, but I have IgM antibodies and no IgG, and I don't have MOG. This is a true negative. Show up, there we go. That's a true negative. So with this assay now, we have much more confidence in this growing spectrum of MOG. So this patient that we tested with Dr. Samuels and many more that I've seen over the past 4 or 5 years has contributed to this growing spectrum of MOG antibody disease as we now know it. So what does the spectrum look like? Well, you've got 2 types of patients. You have monophasic and you have relapsing patients. In the monophasic group, about half of kids with acute disseminated encephalomyelitis will test positive for the MOG antibody acutely. And many of them will remain positive for a few months afterwards, and then they'll go seronegative. That's pretty typical for MOG. And actually that portends a good outcome. So as I'll show you, one of the good things about MOG is that it causes a lot of damage, but functionally patients return to a good level of function. We also see monophasic cases of optic neuritis and transverse myelitis. I just saw a young woman who had an acute infectious mononucleosis with Epstein-Barr virus, PCR positive in the CSF. A few days later, ascending myelitis, tested for MOG antibodies, really high levels. She recovered, made a pretty good functional recovery, and then the MOG antibodies went away, and she's been monophasic ever since. So this monophasic form of MOG is a well-known phenomenon. For whatever reason, these patients tend to do well, and if they lose their MOG antibodies over time, it's a pretty good indicator that they're going to be monophasic. No guarantees. There's a study that said that 15% of people still might relapse if triggered by a potent vaccine or an infection or something like that. So there's no guarantee that you'll remain monophasic. But the other group of people have a relapsing form. And this is the type of MOG antibody disease that's been termed, I hate this name, it's called MOGAD, MOG antibody disease, MOG antibody disease. And the British have named it MOG antibody associated disease. So they stuck an extra A in there, made it even worse. And this disease is now what we recognize as mostly optic neuritis. 66% of the time, we calculate the chances of an attack is going to hit the optic nerves. And when I was in medical school, the big deal with bilateral optic neuritis was it's likely to be neuromyelitis optica, because relative to MS, neuromyelitis optica is much more likely to cause bilateral optic neuritis, but MOG even more so. Okay, probably a quarter of attacks are going to be bilateral optic neuritis. And the lesions are long. The gadolinium enhancement, it tends to be long. We calculated at least 17 millimeters long. That's almost half of the length of the optic nerve or longer, whereas MS lesions tend to be much shorter. And then an interesting phenomenon with MOG is this perineuritis feature. The meninges around the optic nerve tend to be targeted in MOG, and it's really painful, especially when the inflammation is in the intracanalicular region of the skull. And my colleagues in Britain insist that if you have a patient with optic neuritis where pain is a major component of their disease, MOG is a likely offender. Now, when patients come in and they lose vision in one eye or both eyes in the case of MOG or NMO, the attack is usually pretty severe. And when they're in the hospital before their antibodies come back, I don't know if it's going to be MOG or aquaporin 4 NMO, which is known to be a severe variant, known to be a severe disease. But if their antibodies come back before they get discharged, I feel fairly confident telling MOG patients, look, you're going to go from 2200 probably back to 2040, or maybe even better, because MOG patients tend to have really good recoveries. Not everyone does, but every attack tends to make much more recovery than, say, an aquaporin 4 NMO patient would. Now, even though functionally they see better, if you look at their optic nerves and you measure the thickness of the retinal nerve fiber layer in the retina, this is where all the axons from the ganglion cell layer course into the optic nerve. If you measure the thickness of that layer in a MOG patient after a bad optic neuritis, probably half of the axons are gone. And yet they still have a good functional recovery. Whereas an aquaporin 4 patient may lose half their axons and they don't make nearly as good of a functional recovery. So that's another feature of this growing spectrum of MOG that we recognize. This good recovery is also true in the spinal cord. So attacks in the spinal cord can be long, can be small, can be lateral, can be central. And the recovery, again, is really good. I had this one young lady in law school who came in completely quadriplegic. And a month later, rolled into my clinic, still quadriplegic, tested positive for the MOG antibody. And in that case, I was wondering, should I even say anything about prognosis? How can I feel confident that this person who's been quadriplegic for a month is going to make a good recovery just because she tested positive for MOG antibodies? In a year, she was walking again with some assistance, but still walking, which is not usual for most causes of transverse myelitis. So good recovery is the feature here. Then there's this other weird thing with teenagers who test positive for MOG. These are folks who will be sitting in your clinic, talking about high school, boys, girls, whatever, and all they tell you is that they have a seizure once or twice a day. But you get a brain MRI and you see this huge, ugly, enhancing lesion. It's cloudy and white matter, gray matter, and you think, wow, this looks really bad. And then you treat it, they get better. That dissociation between that clinical presentation, the MRI and MOG, that's a known thing in teenagers. Okay, and one more cool thing about MOG before I move on to the next short story is this phenomenon about MRI negative myelitis. So when I was growing up, I would see these patients who would tell me that their story would fit perfectly. You'd localize, Dr. Samuels, if you had to localize these patients, it would be spinal cord. There'd be no other explanation for their scenario, but you scan their spinal cord and you see nothing. And we used to call these cases MRI negative myelitis, but what ended up happening in many of these cases is they'd call their doctor would say, come in 2 weeks, they'd come in, they get examined. Yes, we see the toe going up, you're limping, let's get an MRI. 2 weeks later, they get the MRI and it's negative. Why is that? Because if it was MOG, these patients, like I said, they tend to heal well and their MRIs even normalize. So you might miss it. So if you see an MRI negative myelitis, it could be MOG. And remember, even if they come in 2-3 years later and tell you this story, it could have been a monophasic form of MOG. So MOG is high on our list now for this MRI negative myelitis. So how did this bet end? Well, Dr. Samuels kindly agreed to host a lobster party at his house for the residents, and I'm going to host a party for residents with crabs, but we'll have to wait till the summer because a truism in Maryland is you never crack a crab inside your house because you get pieces flying everywhere, and your house will smell like Old Bay for 3 months. So it has to be outside in the summer. Okay, so a bit more about me. I told you I went to Baylor. This is Baylor College of Medicine in Houston. My wife and I both did MD PhD there, and then we matched at Johns Hopkins for neurology internship, then neurology, then fellowship. And we stayed on the faculty long enough to see the construction of the new building at Johns Hopkins. And this is where I started my career. And I'll say one thing about Johns Hopkins, a really good place to get started as young faculty. They stuck me in an old building with, yes, burnt orange cabinets. And the drawers in this lab, if you open them, you'd have a really hard time closing them. They're decades old, but it doesn't matter because what your lab looks like or how it functions is not nearly as important as the resources, the people around you, the mentorship. And the work we were doing there is the subject of story number 2, which I call proof of concept. So I introduced MOG. MOG is on the end of the severity spectrum for CNS autoimmune diseases on the good side. If you had to have a disease like that, you want MOG. The other end of that spectrum is neuromyelitis optica. So this is my patient, this young lady here. Was on the cover of the Baltimore Sun sports page. She was a star guard for the Dunbar High School basketball team. And she came in with optic neuritis. And at the time she came in, we didn't really know how to treat these patients well, especially kids. And she had repeated attacks. She eventually became blind, triplegic, and passed away within just a few years. And back in these days, the mortality rate for this form, this neuromyelitis optica, that's the aquaporin 4 variant, the mortality rate was up to 30%. Okay, so these folks had really severe disease. They did not make good recoveries from each attack, like MOG patients do. They have a much more severe course. It's not the rule here. We have lots of patients who do well. We have 3 FDA-approved treatments now for this form of neuromyelitis optica, so it's not this dreary. In fact, we ran a new mortality rate study and came up with only 7%, down from 30%, 32%, so that's good. We've made improvements. And the aquaporin 4 antibody, which is associated with this disease, also uses a cell-based assay. Take a hex cell, you express aquaporin 4, add the patient's serum, use a secondary antibody, detect it by flow or by microscopy. And since we have clinical criteria for NMO, I can tell you that this test is 99.99999% specific. The only reason I don't say 100% is because I'm sure somewhere in the world, someone has a case where they think they have a false positive, but I've yet to see one. I had one case of a woman who called me in a panic because after she fell in a park and hit her head, taken to the emergency room, had her blood tested for aquaporin 4, it came back positive. And I thought, wow, I finally met a false positive case. And she had optic neuritis a year later. So she'd already broken tolerance to her aquaporin 4 protein. She had antibody circulating, and it was just a matter of time. So I'm comfortable saying 100% specificity. And the 2 targets now that we have for autoimmune CNS disease are related in the fact that both of them have this upstream immunological process going on. They both have, so MOG and aquaporin 4 have B cells and T cells that are conspiring to attack. They don't live in the central nervous system. In the case of MOG, probably in the spleen. In the case of NMO, we don't know, maybe in the blood, maybe in lymph nodes. But these B cells and T cells, and maybe even farther upstream monocytes, are all conspiring and planning the attack. And then once they decide they're going to initiate the attack, they send their effector cells to break down the blood-brain barrier, and then the rest of the immune system comes in across the blood-brain barrier. In the case of MOG, it binds myelin on oligodendrocytes. In the case of aquaporin 4, it binds astrocyte and Phe. And this immune attack is very, very specific. Patients who have antibodies to aquaporin 4 on the astrocyte end foot has lots of other proteins there with it. Potassium channels, it's bound to agrin in the basement membrane. But people with NMO don't have antibodies to those targets, just aquaporin 4 and just MOG. So it's a really, really specific disease. And the value in having a very, very specific immunological target is that you can apply immunological principles to try to correct the imbalance. So when you're born, the job of your immune system is to decide what is self and what is not self. Whatever is self, you want to build a tolerance to. You don't want autoreactive immune cells going around your body picking off your own cells, okay? But you do want active immunity against foreign proteins. So this balance is really critical to establish what's self and what's not self. Patients with aquaporin 4 and MOG, they have very, very specific defects in this balance. They think that aquaporin 4 or MOG is foreign, and they're mounting an active immune response to it, and they are not tolerant of it. They weren't born that way, though. They acquired that intolerance at some point in their lives, maybe after an infection, we don't really know. And there must be a genetic susceptibility to even allow that. But that's the imbalance that we're trying to correct. Now, how do you model this break in tolerance in mice? Well, mice are born with an innate tolerance for their own MOG. They know MOG is self when they're born. Okay, they're not usually going to develop a demyelinating disease. but you can induce it. You can break their tolerance to MOG by injecting that MOG protein with a whole bunch of bad acting proteins like Mycobacterium tuberculosis proteins, for example. So if you take a mixture of Mycobacterium proteins, put MOG with it, inject it under the skin, give a healthy dose of pertussis to really activate the immune system of this mouse. The mouse's immune system will think that MOG, associated with all these bad proteins, might be foreign after all. And it'll mount an immune response against its own MOG and develop an ascending paralysis that looks, again, like demyelinating disease, as I mentioned before. This is what the typical course would look like for a mouse with this break intolerance to MOG, which is called experimental autoimmune encephalomyelitis. After a few days of this break intolerance, the T cells will invade and the mouse will get sicker. So 1 is a limp tail, 2 is a partial hind limb paralysis, 3 is complete hind leg paralysis. You can see over time, the mouse gets weaker and weaker, and then it just sort of levels out. If you add corticosteroids, as we would for a patient, or in this case, they tested FTY-720, which is fingolimod, which was ultimately approved for MS. Any way that you want to suppress the immune system, you can tamp down this immune response against its own MOG protein. So this is how you break tolerance in a MOG. So now this mouse thinks that MOG is foreign, or at least associated with a bunch of foreign bad actors, and is going to attack MOG as well. And this is the model in which you can develop a tolerization therapy. Because if this mouse broke tolerance to MOG using a very specific protocol, can you unbreak it? Can you return it, switch the immune system off to this very, very specific protein? So this approach is being trialed by none other than BioNTech. This is the company you might have heard of, who partnered with Pfizer to make the COVID vaccine. Many of you probably got the Pfizer vaccine using their technology. What they specialize in, what BioNTech specializes in, is creating these lipid nanoparticles. And they can target where they want their lipid nanoparticles to go and how immunogenic the RNA inside of it is. That's their specialty. What they published a couple of months ago was that they could take a new lipid nanoparticle they developed, stuff it with MOG RNA, and deliver it into mice with EAE and switch off that immune response to MOG. Okay, so what they did was they established the same EAE model I just showed you with MOG, and then they added their special lipid nanoparticle by injection with their MOG protein into the mouse. And in contrast to the controls that got sick, the mice that got their product, their lipid nanoparticle with MOG, their MOG vaccine, did not develop any disease at all. Even if they injected their mice after the mice got sick, they could still blunt that response a little bit. So how did they make this special lipid nanoparticle? Well, what they claimed is that all they had to do was craft their lipid nanoparticles with some slightly negatively charged lipids, and it would target the particle to the spleen. In previous papers, they showed that you can manipulate these nanoparticles however you want and target them to different organs. A slightly positive charge, for example, goes to the lung. So these negatively charged particles go to the spleen. As I mentioned with MOG, the spleen is the room where the decisions are made for MOG antibody disease. So that's why they targeted the spleen, and they created a non-immunogenic RNA for MOG by methylating the UTPs. So when the lipid nanoparticle got to the spleen, antigen presenting cells picked it up. They figured that this non-immunogenic MOG was harmless. So they got a harmless signal from this lipid nanoparticle. And then they told all the T cells in the spleen, okay, you know what? MOG is not bad. Let's take it easy on MOG. So you could demonstrate that by taking all the T cells out of the spleen and showing that more than 80% of the T cells after this vaccine are of the regulatory harmless type that test positive for this marker called FOXP3. So 80% of the T cells now are the harmless regulatory type. And it turns out if you establish a ratio of about 80% regulatory T cells to 20% pathogenic T cells, that's enough to calm the immune system down. So they didn't do anything to the bad T cells with this lipid vaccine. All they did was they created a regulatory component to try to restore the balance to this immune system. Okay, so how do you do this with aquaporin 4 for neuromyelitis optica? Well, in contrast to the EAE model with MOG, it's really hard to convince a mouse that aquaporin 4 is foreign. A mouse that's born with aquaporin 4 is pretty convinced that aquaporin 4 itself, no matter what you do it. You could put aquaporin 4 into a mouse with the worst bad actors you can imagine infection-wise, and it'll never confuse the two. It'll know aquaporin 4 itself. You really have no ability to break tolerance to aquaporin 4 in mice. So we went around that problem by using knockout mice. Okay, knockout mice that don't have aquaporin 4, they don't know that aquaporin 4 itself. They think aquaporin 4 is foreign. And so then if you take aquaporin 4 proteins, like especially this one that we found was particularly pathogenic, you inject that into a knockout mouse, that mouse is going to mount a vigorous immune response to aquaporin 4 as if it were foreign, because it is to this knockout mouse. Now it's harmless in this knockout mouse because there's no aquaporin 4 to attack. But if you take those T cells out, if you take the B cells out with it, the antibodies with it, inject it into a wild type littermate, now these mice have aquaporin 4, and these T cells still think that aquaporin 4 is foreign, so they're going to attack. So that's the model we use to break the tolerance in aquaporin 4. And this is what it looks like. Same type of EAE as MOG, where after a few days, those T cells invade and it causes weakness. The other really cool thing about aquaporin 4, the aquaporin 4 model, is that it causes a pretty robust optic neuritis as well. And I've been working with these black 6 mice my whole career, and they have these beady black eyes, and I've never actually seen them blink until we established this model. They have these fluorescent purple eyelids, and they blink like every 3 seconds with this optic neuritis. And that was one of the first things we noticed after we created this model. So these are T cells in the optic nerve. You can see how invaded it is with secondary demyelination. So you get transverse myelitis, optic neuritis, all induced by aquaporin 4 reactive T cells, very much similar to the human disease. So this is the model now that we're targeting with tolerization. Now, I mentioned how BioNTech is approaching their MOG vaccine is by generating regulatory T cells. This approach is called suppression. You're not doing anything to reduce immunity to MOG or aquaporin 4. You're just creating regulatory T cells to suppress that immune response. Now, we could do the same thing. We're not experts at lipid nanoparticles, but you could do the same thing with desensitization. Desensitization is an old method that people have used for decades to get over their immune reactions to things. Desensitization also works with allergies. So farmers who would get an allergic reaction, a rash to hay, they found that by chewing on hay, they could tolerize themselves and not get the rash. My lawn keeper chews on poison ivy, so he doesn't get a reaction, poison ivy. You can desensitize to drugs, you can desensitize to a lot of different things. And so we're desensitizing to aquaporin 4. And the way we do this is we take those knockout mice, and after we induce that robust T cell response, then we start to desensitize it, to build that regulatory T cell against aquaporin 4 to try to restore some balance to that seesaw against aquaporin 4. And how does that work in the mouse? This green curve is what typically happens in a normal NMO mouse, where they get sick and they recover. Then you induce a relapse, for example, just by injecting more aquaporin 4 protein, you rev up those T cells again, and it causes a worse relapse. So this is the mouse without the vaccine. With the aquaporin 4 vaccine, this includes all those regulatory T cells trying to balance that immune response. You get a much more subdued response. Even with the relapse, it tends to be more subdued. Now you have to keep boosting those aquaporin 4 regulatory T cells. If you don't do it on a regular basis, you will lose that regulatory response. There was one summer in Maryland, I got swarmed by wasps, and I woke up in the emergency room after an anaphylactic reaction. They sent me to an immunologist. I started taking desensitization shots. They injected the exact same wasp venom to yellow jackets that I was allergic to, And then I built up a regulatory T cell response to it. And then I stopped using it because, I'm lazy. And what happens then is that regulatory response goes away. Hopefully my immunity goes away as well, but that's the problem with this regulatory approach, is you have to maintain it. Now, what if you don't want to try to maintain it? What if you just want to turn the immune system off completely to these pathogenic T cells? Well, there is one more really high risk approach, and it's called activation-induced cell death. This is going to sound crazy, but imagine if after that wasp attack, and I was swarmed by these yellow jackets, I still remember it. What if at that moment, right before I passed out, you said, okay, I'm going to inject you with a lot more venom in a really highly soluble form, and I'm going to convince your T cells, I'm going to overactivate them and kill them before they react. So that's what activation-induced cell death is. You give the exact same antigen that they're about to react to, but you give it in such a high amount that you literally overactivate T cells. T cells that are activated have to rest. So they're active and then they rest, and then they're active and they rest. And they have this very strict time protocol for that resting period. If they don't rest, they die. So that's the idea behind activation-induced cell death. Now, it's a little bit risky. You're not going to do this in people with a wasp allergy just yet. But in mice, you can try it out. And this is what happened when we gave what we're calling the aquaporin 4 antidote. So these mice have the T cells that are about to invade. And then we literally overwhelm them with a really high dose of the same protein that those T cells are reacting to. And look at what happens. They have absolutely no disease. And what sounds really good is tempered by the fact that about 20% of the mice had an acute reaction and died within a few hours. So about 20% of mice just had this horrible immune reaction and passed out. But if they survived, then they had no NMO. And you couldn't even trigger a relapse. Those T cells were gone. So this is a way to literally get rid of those autoreactive T cells, but it comes with this caveat that you might die. So this is not exactly something that I said we're translating to the clinic. That's another, maybe the title for this short story should have been not quite ready for clinic, but it demonstrates the power that we have now to manipulate the immune system. We can literally turn it on. You can develop MOG and aquaporin 4 disease, and then you can turn it off. This is what we're working on currently in the lab. Okay, so when I came to Mass General, they set up a lab for me in this building. This is building 114 in the Charlestown Navy Yard. This is the water view. Okay, this is not the view that I have. This is Rudy Tanzi's view. This is not my view. I look out on the street. As I said, it doesn't really matter what your lab looks like or what it looks over. It only matters the environment that you have, which is, again, a hugely rich environment with lots of wonderful people. And I'm really starting to stick my finger into a lot of different pies at Mass General. On the research side, working with Brian Baxke on the role of aquaporin 4 in the lymphatic system, working with Michael Schwarzschild and Zhixuan Chen on the role of the immune system in Parkinson's. If you're wondering what the role of the immune system in Parkinson's is, you're right to think that way, but there is. We're doing a biomarker screen with the Lurie Center in autism. That's really cool. We have a program for stem cell transplantation in my lab into optic nerve and spinal cord. We're doing lots of trials in NMO and MOG. Mentioned we already have 3 FDA-approved trials, FDA-approved drugs in NMO, and we're launching trials in MOG as well. On the clinical side, if you have any cases that are weird, unusual, head scratchers, they're probably autoimmune. Go ahead. I love those cases. I work with the Harvard Undiagnosed Disease Network, and that's a really cool group. We'll do a lot of genetic analysis on them as well. So if you have any reason, any interest in these areas, please do contact me. Okay, my final short story is on familial transverse myelitis. And this transverse myelitis is not the same transverse myelitis as I described in MOG and in NMO. This is a different disease entity. This is a monophasic process. This is an immune-mediated attack of the spinal cord that causes some destruction, neurological dysfunction, and then it goes away. Usually follows an infection after 3 weeks or a vaccine after a week or two. And as I mentioned, all of these cases by definition are monophasic. So I had the opportunity to meet two sisters with this condition. I just want to mention transverse myelitis is pretty rare. This monophasic form, maybe 1 in 500,000, something like that. You might have heard of the cases in the AstraZeneca COVID trial, a case in reaction to their COVID vaccine and one in reaction to the meningococcus vaccine in the same trial. That's the type of transverse myelitis we're talking about, but it is a rare condition. So to have two sisters with it was very unusual. The first sister had her attack a long time ago. She's in her 50s now, but her onset was at age 15. It was acute, sensory motor, lost her bladder function. These were the days before MRI. She insisted she had a pleocytosis in her CSF. I called the hospital. Hospital has been long closed. She got some steroids eventually, but did not make a great recovery. Took her about 2 years to use a walker, but she used a motorized scooter for most of her life. And 35 years after her attack, she got an MRI, which showed a subtle lesion here. You could see it probably better on the axial cuts around T9. And she's still very much affected, but has been monophasic since that attack. So her sister called her when her sister was 50 years old, called her one day and said, I think I'm having the exact same thing you had when you were 15. I can't really move my legs. I can't feel them well. I can't pee. And her sister said, go to the hospital. You need to get steroids on day 1, which she did. She got the MRI, which showed just a subtle lesion, also thoracic cord, one blip on the axial cut, which is right here. It really convinced them that that's what this was. She got a course of steroids and it melted away and all of her symptoms resolved. And they came to me and they said, could this be genetic? We have 2 brothers. Are they at any risk? And I said, this is a great opportunity for a genetic study. So I called up, there's this Johns Hopkins collaboration with Baylor funded by NIH to sequence families that have these rare conditions. And they accepted this case and they sampled the 2 brothers who were healthy and then the 2 sisters who had transverse myelitis. And they called me after about 6 months of this. And they said, well, we found an autosomal recessive homozygous mutation in this gene called VPS37A. Does that mean anything to you? And I said, no, I have no idea what VPS37A is. I'm imagining Craig Blackstone, our new division director for movement disorders, is probably shaking his head because VPS37A is an important protein, as I'll show you in other movement disorders, but I had no idea what it was doing in transverse myelitis. But the genetics folks were interested in it because this change from a C to an A changed the leucine in this protein to an isoleucine, which was predicted to be deleterious, which they said was unusual because almost every other mammal had a conserved leucine at this spot. And they thought, this has got to be meaningful. Does it make any sense to you? And as I said, I don't know, maybe. It's a very rare variant. In the databases, only 1 in 1,000 is even a carrier. So you'd expect a frequency of 1 in 4 million in the general population. But it turns out that when you look at the databases, there's none. Not a single autosomal recessive homozygous person has been recorded in any database worldwide. So I thought, okay, this is kind of interesting. I still haven't looked up VPS37A. I'm kind of sitting on these 2 cases thinking one genetic illness, maybe just restricted to these 2 sisters. What we did was we collected saliva samples from patients who would come to our transverse myelitis patient days. So on Saturdays, once or twice a year, we would host a patient day, an educational event. Patients would come in, they'd learn a little bit about TM, and in exchange, they'd spit in a tube for me, and we'd extract their DNA, and thank you, Santi, for doing all the sequencing. And what we ended up finding was another woman with transverse myelitis who had this exact same mutation, the exact same exon, exact same everything. And her story was that she was 51 at her onset, acute onset sensory motor pain. Her MRI was done in Canada. It's a little bit more obvious compared to the other 2 sisters. You can see the lesion here, also thoracic. And she responded very well to a course of steroids. She recovered her ability to walk. she still has some pain, but that's her only residual issue. And so now we have two families with transverse myelitis, the two sisters and then the new patient here, and carriers, one brother carrier, and she came with a sister who's a carrier. And the fact that we have now two families and every single person in the database with this autosomal recessive mutation has transverse myelitis just made us think maybe there's something to this. Let's look into it. So what is VPS37A? VPS37A is part of this SCART system. So every membrane protein gets taken up once in a while, brought into the cell to be interrogated and either recycled or put back out on the membrane. And what VPS37A does is it helps bring that membrane in and localize it to the right place. There are many proteins that do this. And this whole system is called the SCART vesicle recycling system, and there's many different components to it. Here's VPS37 in yeast. In mammals, you have 4 isoforms, A, B, C, and D. So it's part of this huge group of proteins. And VPS37A is expressed in almost every cell we looked at. Here is colonic epithelium, even glioblastoma cells, the green dots, have VPS37A. It's not restricted to the spinal cord. And SCAR proteins have been associated with other neurological diseases, including Alzheimer's, degenerative disease. And then really interesting to us, there was a previous report from Israel with 2 Palestinian kids who had what they called spastic paraparesis with a different mutation. So ours was the leucine in 234. This is a lysine mutation in 382, which they said caused a neurological disorder. And they modeled it in zebrafish. They knocked it out and found that the zebrafish didn't have a back fin that could move. And they attributed it to a spinal cord dysfunction. We don't know if those kids were born that way or if they developed transverse myelitis, but again, very, very similar phenotype. And so how could a genetic illness contribute to a one-time event? In 2 cases, it happened in their 50s. So a genetic illness that causes a transverse myelitis in their 50s, what could go wrong after 50 years? Well, there is a growing list of genetic illnesses, genetic mutations that can lead to autoimmune disease. The most obvious one is this autoimmune regulator gene, which is responsible for educating your immune system. So obviously the problem in that gene, you get a whole host of autoimmune problems. But what I think VPS37A is going to do is fit into this idea of a CNS infection risk or CNS myelitis risk. So the classic example is a TLR3 mutation, which predisposes to a herpes simplex virus encephalitis. So people who have this mutation are much, much more vulnerable to a herpes simplex encephalitis infection. And maybe people with this VPS37A mutation are more susceptible to name the infection that then causes myelitis. It may just be a genetic predisposition to an insult from the environment. So we got a small grant. We sequenced more parts of VPS37A around exon 6. We included more exons, and we found more mutations clustered around exon 6. Now we have some compound heterozygote mutations in exons 3 and 10, a lot more mutations around this exon. And so we decided to model it in mice by knocking it out. And initially what we're finding is that it seems to be lethal. So if you knock out the gene, the mice aren't born. We have to do timed matings to really figure out when this defect is. But as of right now, it seems to be lethal. So we're making conditional knockouts. These mice are born with VPS37A, they develop, and then we inject them with tamoxifen, and then it gets knocked out. And then we'll be able to see if it predisposes them to whatever insult. So you'll have to invite me back in a year or maybe 2 years to give the rest of that talk. But all this work has been done by people in my lab, listed here on the left, and my kids on the right are the people who keep me from doing the lab work. And thank you for your time. Happy to take any questions.
Thanks so much, Dr. Levy. I know not everyone in the Ether Dome can hear, but There's one question in the Q&A. I'm happy to read it, or I can let you read it so that folks that are there with you can also hear.
Okay, Dennis asks, how often do children with sickle cell disease develop MOG-associated disease given they grow up with functional asplenia? Great question, Dennis. I haven't seen a case yet. If you don't have a spleen, does that mean that you're resistant to MOG antibody disease? Does that mean that if you don't have the organ where the disease is hosted and where it's sort of brewing to begin with, does that maybe make you immune? Maybe. I haven't seen a single kid with sickle cell disease develop MOG. So what does that mean? I don't know. We'll have to collect more cases. That's a great question, though. Marcelo.
Well, excellent talk. I was even thinking how much you could fit in 1 hour in the scope of your work from the clinical translation and towards the lab course. Congratulations.
Thank you, Marcelo.
All your discoveries. It's a dream come true to have you join us at MGH and MGBs.
For those of you on the call, Marcelo is flattering me very much. I love it. Thank you, Marcelo.
Question is, what was the response to steroids? Tell us about the pathogenesis of the MOG in comparison to F.4, myelitis, optic neuritis, or even MS.
Yeah.
What do we conclude about that?
What Marcelo is asking is a really good question about the steroid sensitivity of MOG patients. This is another really unique feature of MOG. They respond very well to steroids, so that if they do have an acute attack and you give them steroids, they tend to recover really well and at least will abort that attack. You often can defer plasma exchange in favor of just continued steroid use because they often respond very well. But what happens if you stop the steroids, either abruptly or you taper too quickly, then you often get a steroid withdrawal relapse. And it'll usually happen within a week. So if you give them their 5 days and then you don't do the taper, within a week they'll call and they'll say they'll have eye pain again and they'll be coming in with a relapse. And that steroid sensitivity is very interesting. It's much more unique to MOG than it is to NMO and certainly to MS. It's kind of like on the sarcoid lymphoma type of thing where it's exquisitely sensitive to both treatment and to then withdrawal.
Do you think that's due to the target, that we have no time to stop the further downstream of inflammation?
Yes, so what is that mechanism? I don't know. It's a good question. We generally will taper them off slowly, and that tends to avoid a relapse. So why does that happen? I don't know. There are glucocorticoid receptors in almost every immune cell, and they all are responsive to steroids in some degree or another. And what happens when you withdraw that steroid? What does that then do to that immune? I don't know. That's a very good question. Please, Dr. Samuels. Thank you.
Let me ask you a two-part question. The first one is just very
Okay, this is a two-part question. That is a great question. So the question is, can a skilled neuropathologist look at a biopsy or autopsy tissue of MOG and be able to distinguish it from, say, multiple sclerosis? I'll give you an actual anecdote. So one of my patients was biopsied before the MOG antibody test became available. And I got a call from a very skilled pathologist who said, this is MS. I don't care what you're telling me about the MRI and her clinical course. If you don't treat her for MS, it would be malpractice. And I just felt like this was not MS clinically, even though by pathology it was. Turns out, a year or two passed. We did finally test her for MOG. She tested positive. I called the skilled pathologist back to tell her the results of the MOG antibody test. She said, oh, yeah, we already knew that. We found out that she was positive. We now know what MOG antibody disease looks like. And it looks a lot like MS. Now, there are some, there are 4 different pathological phenotypes of MS, depending on what stage the lesion might be in or what patient might develop it. So there's a little bit of heterogeneity with MS. And with MOG now we have probably, I'm guessing, 25 cases in the literature that have been published with a little bit of heterogeneity. No, not the amount of complement that you see with aquaporin 4 NMO. You might see a little bit on the rim, like with MS. The pathology generally shows demyelination with macrophages that looks much, much more like MS compared to NMO.
So the second part of the question is this, I'm interested in where these diseases were for Europe. Some of them we have theories about where was channel for the system paralyzed in this hospital.
Yeah.
Saw it.
Yeah.
Where was the central quantum myelitis or saw in the cases? Where were those cases? Were they capital German very, very skilled?
Yeah.
The whole way. It didn't mix it up with QMS.
Yes.
The bigger question is, how does one define a disease? Do you find a disease by test or syndrome or
so yeah, so Dr. Samuel's second question is, where were all these MOG patients before the MOG antibody test? What were they, where were they, and what were they being mixed up with? And the answer is they were in the MS clinic or the NMO clinic or the ADEM clinic. So they were called recurrent ADEM. In mass eye and ear, they were called chronic relapsing inflammatory optic neuropathy. In my clinic, I called them seronegative NMO. And in MS, they were considered intractable or not, poor response to treatment in MS. And it wasn't until this MOG antibody became available that they all started coming in under the appropriate umbrella. And as a neuroimmunologist, the answer to your second part of your question, which is what is the disease really? I think it really is, in the case of these MOG patients, an immunological reaction to MOG. I think that's the best way to define it. We should call every antigen-specific disease by their target. And I think what's going to happen with MS, where you have this heterogeneity, is we're going to start picking it apart. They're going to be the folks who have pathology, type 1 being segmented off when we can identify them by blood test, and type 2 and so on. They'll all be segmented off. And I think they'll all have a different immunological phenotype. And so MOG is in the heyday right now as patients are being tested and recognized. There'll be another antibody next.
I have 50 more questions.
There are a few questions in the chat as well, Dr.
Oh, okay, right.
So, Doctor, there are more flattering comments if you care to read them. But Dr. Sikovic also has a question.
I can ask it. Well, I gave some flattering comments. I'm so excited that you're here, and you've been such a short time. I was just curious about your comments about T regulatory cells, that basically you have to continuously give them for them to be helpful. But that is actually being done in some neurodegenerative diseases. I was just kind of curious if that was a MS strategy, a real MS strategy, or demyelinating strategy.
A lot of MS trials have been done to create. So what Dr. Sikovic is asking is, can you generate regulatory T cells as a strategy to treat MS, as they're doing for other degenerative diseases? And the answer is, it's been tried many, many times. People pick out many different parts of myelin and CNS, and they add in some astrocyte proteins just to be safe. And people do generate regulatory T cells to those specific antigens. But what happens is, in MS, they shift, the antigens shift, and they start attacking different parts of the central nervous system, different parts of myelin. So they do retolerize to those parts of of myelin that they are immunized against with this vaccine therapy, but they continue in their disease phenotype because of this antigen spreading phenomenon. Aquaporin 4 and MOG, we think, are going to be better models for this proof of concept because they don't have antigen spreading. It's really a restricted disease. That's a great question.
Thank you.
There's also a question in the chat I'll read. And it's already 10 o'clock. I'll go through this really quickly. Can you say more about biomarkers in autism that you're investigating? This is someone who worked with Carlos Pardo at Hopkins, who is a neuropathologist and is very interested in autism as well. And what we're looking for are biomarkers against CNS targets in autism early on in the course of disease, where we think the immune system may play a role. There's some tantalizing data that the immune system could be involved in autism, and that's what we're looking into. And Andy, I can go into that in more detail with you offline.
If you have 1 more minute and can answer, there's another question from one of our residents, Giovanna Manzano, under L6 workroom.
Do I see that here?
In the chat, I can just read it for now. What is your general approach regarding when to start disease-modifying therapies in patients with MOG beyond steroid monotherapy? Is it just one clinical or radiographic recurrence alone? And separately, is there any utility to following OCT in these patients to guide management?
Great question. So with MOG, there's a large population of monophasic disease, and the general inclination in the field is not to start preventive treatment unless you're sure they're in the MOGAD relapsing group, because you don't want to use lifelong or long-time immunotherapy in somebody who doesn't have a risk of relapse. Now, how do you know for sure? If you test positive for the MOG antibody over and over again, sustain positive for 6, 12 months, 18 months, the Mayo insists that you have an 80% risk of relapse. It's reasonable if you've only had 1 attack, especially if you've had a severe attack and you don't want to risk it, to then go on immunotherapy. But otherwise, if you've had 1 attack, you've recovered well, your MOG antibody went away, we don't treat with preventive therapy. And was that?
And then the second part was, is there any utility to following OCT to guide management?
Question about OCT to guide management. With MOG, we think the attacks occur as overt relapses and not subtly as a degenerative process like it does in MS. So we think the OCT should be stable in between, during remission, in between relapses. And OCT is really helpful when the MRI is not helpful anymore. So a patient who has really, really thinned optic nerves from years of attack, if they come in with pain, even if they can't see well in that eye, it's usually the OCT that's helpful in guiding management, because the MRI is just, it's too small to see the optic nerve anymore. Great question.
Wonderful. Thank you so much.
Thanks, everybody.
Taken extra minutes of your time, but this was wonderful to hear about your experiences and some of your personal journey as well. So thanks for joining us.
Thank you, Priya.
We'll indeed have to have you back in a couple of years.